Hello everyone, I am Duel Bose. This is the second lecture in the 4-part series of Demystifying Genetics. This session is specifically meant for high school students, undergraduates, or even doctors who wish to brush up their genetics. In the first part, we have already covered the basic definitions like nucleotide base pairs, DNA, chromosome compaction, central dogma, etc. Today's session is specifically dedicated to cytogenetics, which is the study of the structure of chromosome. We will talk about chromosomal errors, Ploidy errors, numerical errors, and so on. The contents of a chromosome. We already saw in the first lecture how chromosomes were compacted. So, what do chromosomes contain? Chromosomes contain DNA and packaging protein. Approximately 40% of chromosome is dedicated to DNA, which contains coding as well as non-coding genes. However, 60% of the chromosome is packaging protein, which is made up of histones and structural proteins. Now coming to the chromosomal states. Heterochromatin is inactive chromatin, whereas euchromatin is actively involved in RNA transcription. Heterochromatin appears denser than euchromatin under the electron microscope, and it stains darker under the light microscope because it is highly condensed. Eu in Greek means good, like eulogy, euphony, so on. Hence, euchromatin is the active chromatin. So when the chromosome is stained by Giemsa or G stain, we typically see dark and light bands called G bands on a chromosome. The darker bands are heterochromatin, while the lighter bands are euchromatin, representing the active gene. Some key terms before we move forward. You must have heard homologous chromosomes as well as sister chromatids. Homologous chromosomes are chromosomes of equal length, equal position of the centromere, and have similar gene on their corresponding loci. We inherit one from our mother and one from our father. After replication, the homologous chromosomes are joined at the centromere and the arms are called sister chromatids. This is a typical structure of a chromosome where the tail ends are called telomere joined by a centromere, and the centromere divides it into two arms. Chromosomes can be represented by either structure on your left or the structure on your right. What you see on the left is chromosome at prophase represented by a single strand, while what you see on the right is a chromosome at metaphase where chromosomes replicate right before cell division and appear like an X. The two replicated strands are called sister chromatids, which are connected at the centromere. The centromere divides the chromosome into two parts, that is the P arm and the Q arm. P arm is usually the smaller arm or petite. The spindle fibers attach themselves to the centromere and pull one copy each of the chromosome apart during mitosis. The chromosomes can be classified into 4 subgroups on basis of position of the centromere. So the position of the centromere can differentiate chromosomes into 4 types. Metacentric chromosomes have centromeres almost centrally placed. This divides the chromosome arm into almost 2 equal parts. Human chromosomes 1 and 3 are metacentric. Submetacentric chromosomes have the centromere slightly offset from the center, leading to slightly shorter and longer arms. Human chromosomes 4 through 12 are submetacentric. Acrocentric chromosomes have a centromere which is severely offset from the center, leading to one very short and one very long arm. Human chromosomes 13, 15, 21 and 22 are acrocentric. Telocentric chromosomes have the centromere at the very end of the chromosome. Humans do not possess telocentric chromosome, but they are found in other species such as mice. Another way of classifying chromosomes is by autosome and sex chromosome. Humans have 22 pair of autosomes numbered from 1 to 22 arranged in descending order of the size of the chromosome. That is, chromosome 1 is the largest and chromosome 22, well, chromosome 21 is the shortest. The numbered chromosomes are autosomes which are always present in pairs, one from the mother and one from the father. Autosomes are homologous, meaning they have similar gene in the similar order on them. The sex chromosomes are non-homologous chromosomes and present as either XX or XY. This is a typical snapshot of human chromosomes by their banding pattern. This is also referred as karyotype. Remember that euchromatin stains light while heterochromatin stains dark. So karyotype is nothing but a snapshot of human chromosomes. One important information that one should note at this point is that each band, which is represented as a light and a dark band on a karyotype, represents approximately 7 million to 10 million base pairs of DNA, and it may contain several hundred to several thousands of genes per band. This brings us to the chromosomal errors. Now is the time to look in deep into the numerical and structural errors and difference between Ploidy and Somy. Before we move to the classification of chromosomal errors, it is important to note this one concept which we actually covered in our previous lecture. We saw that chromosomes contain genes and gene code for protein. So if we have a missing gene or extra chromosome, the dosage of the protein changes. Let us see how. What you see on the screen are a pair of a homologous chromosome with similar genes coding for similar protein. So this is the normal production of protein where each gene is producing their bit. In case of an extra chromosome or an extra copy of a chromosome, there is an increased production of protein. And in case of a missing copy of a chromosome, there is a decreased production of protein. Chromosomal aneuploidies cause chromosomal instability, where we either have too much of a protein coded if we have one extra chromosome, or we have too less of protein coded if we have a missing chromosome or decreased expression of the gene. This leads to either protein abundance or protein scarcity and causes imbalance in cell, leading to various disorders. Now let us quickly get back to the classification of chromosomal errors. Chromosomal errors can be classified into either numerical error or structural error. Numerical errors are error in the total number of chromosomes. This can be further classified in either having more than 2 complete sets of chromosomes, that is, Ploidy errors, or a mismatch in the total number of chromosomes. We all know that most of the cells in our body are diploid, where we have 2 sets of chromosomes, that is 2n, except for our gametes, which are haploid. There is only one set of chromosome represented by n. So haploid is n, diploid is 2n or 2 sets. Similarly, triploid is 3n or 3 sets. So as we see, triploidy is where you have 3 complete sets of chromosomes leading to a total number of 69 chromosomes. And tetraploidy is 4n, which is 4 complete sets of chromosomes leading to a total 92 chromosomes. Polyploidy is common in plant cells. However, it is important to note that some very critical cells in our body like heart muscle, bone marrow, liver cells and even placenta are polyploid. Approximately 15% of all spontaneous abortions are due to triploidy in the developing embryo. Triploidy happens to be the third most common chromosomal anomaly, and approximately 2% of first trimester miscarriages are due to tetraploidy. The next numerical error is when there is deviation from the total 46 chromosomes, and this is called aneuploidy. So one less chromosome makes it monosomy, where you have total 45 chromosomes, while one more chromosome makes it a trisomy, giving it a total of 47 chromosomes. So trisomy 21 is one extra 21st chromosome. So quick recap, difference between ploidy and somy. Ploidy is 2n, 3n, 4n, that is 2 sets, 3 sets or 4 sets of chromosomes, whereas Zomies are 2n plus or minus 1, which is 46 plus 1 or 46 minus 1. Quickly getting back to the chromosomal anomalies, the next type of chromosomal anomaly is a structural rearrangement. As the name suggests, it is an error in the structure of the chromosome. Structural rearrangements can be broadly classified into two groups: unbalanced structural rearrangement and the balanced structural rearrangement. In the unbalanced structural rearrangement, the total amount of genetic information changes. Either there is loss of information due to deletion, or there is gain of information due to duplication or addition. The other type is balanced structural rearrangement, where the total amount of genetic information remains same, only its location changes. Unbalanced structural rearrangements. Unbalanced structural rearrangement can be classified into four types: deletion, duplication, insertion, or a ring. During deletion, a segment of a chromosome is deleted. This results in a total loss of genetic information. And since there is a loss, this is an unbalanced structural rearrangement. During duplication, a segment of chromosome is duplicated, resulting in total gain of genetic material, and hence it is classified as unbalanced. Now, insertions are complicated. Insertions could be either inserting a small or a large segment of the same chromosome or another homologous chromosome. And there is a reason why they are called unbalanced structural rearrangement, even though it apparently appears that there is no loss in genetic material. And that is beyond the scope of this lecture. Ring chromosomes are unusual circular chromosomes, generally resulting from breaks at the end of both chromosome arms, with subsequent fusion of the broken ends to produce a continuous ring. Due to its circular nature, a ring chromosome will be compromised and unstable at cell division, leading to genetic instability. Balanced structural rearrangements. As the name suggests, there is no net loss or gain of genetic material. Balanced rearrangements are of two types: inversions and translocations. Inversions, as shown in the figure, occurs when a chromosomal segment breaks, reorients at 180 and reinserts itself. And hence there is no net gain or loss of genetic information, and hence it is a balanced one. Translocations can be of two types: reciprocal and Robertsonians. Reciprocal translocations are a transfer of genetic material between non-homologous chromosomes, where segments of a chromosome are exchanged. There is a give and take, and hence no loss in genetic material. Robertsonian translocation typically involves acrocentric chromosomes. If you remember, we just saw what acrocentric chromosomes were, where the centromere is placed at an extreme end. So chromosomes 13, 14, 15, 21 and 22 are acrocentric chromosomes. In a Robertsonian translocation, the long arm of any two acrocentric chromosomes join to produce a single metacentric or submetacentric chromosome. So there is typically no loss of genetic material. However, due to joining of two chromosomes, the total number of chromosomes in such individual remains 45 instead of 46. So this gives rise to both numerical and structural error in case of a Robertsonian translocation. About 1 in 1,000 people have a balanced structural rearrangement of Robertsonian translocation and are not aware of it, as they are phenotypically normal. Carriers of Robertsonian translocation typically come to know about their condition in an infertility clinic. Patients with reciprocal balanced translocation have a risk of either recurrent pregnancy loss or having an affected child and infertility. This almost brings us to the end of this lecture. A few important take-home messages. You must have heard the terms cytogenetics and molecular genetics. I think most of us must have heard about cytogenetic lab or a molecular genetic lab. So what is the difference between cytogenetics and molecular genetics? Cytogenetics is the study of chromosomes and everything and anything related to chromosomes, and the techniques used in the cytogenetic labs are usually karyotyping, chromosomal staining, FISH or array CGH. While molecular genetics is the study of genes and gene mutation and anything and everything related to genes. Techniques used in a molecular genetic lab is usually PCR, molecular cloning, RNA and DNA sequencing. To get a patient karyotyped, you would typically send the sample to a cytogenetic lab. Technically, you can also send it to a molecular genetic lab, but karyotyping typically means a cytogenetic lab. And if you have to get an embryo biopsied to understand genetic disorders, you would have to send the sample to a molecular genetic lab. Some facts about chromosomal disorders, which are fascinating. 1 in 263 people are affected by chromosomal disorders. Down syndrome or trisomy 21 is the most common disorder in a live-born human. Turner syndrome or 45X is the only full monosomy compatible with life. So this brings us to the end of this lecture where we spoke about cytogenetics. The next lecture would be on genes and genetic mutations. Thank you so much for your patient listening. Thank you.